Hello everyone, I'm Mattia Fragola and I'm going to talk about the seasonal changes of the eukaryotic community structure and diversity in PM10 samples over southeastern Italy. So the atmosphere represents an underexplored temporary habitat for airborne microbial communities such as eukaryotes, whose taxonomic structure changes across different locations and/or regions as a function of both survival conditions and sources. A preliminary dataset on the seasonal dependence of the airborne eukaryotic community biodiversity detected in PM10 samples collected from July 2018 to June 2019 at a coastal site representative of the central Mediterranean is provided in this study. Viridiplantae and fungi were the most abundant eukaryotic kingdoms. Streptophyta was the prevailing Viridiplantae phylum, whilst Ascomycota and Basiliomycota were the prevailing fungi phyla. Brassica and Panicum were the most abundant Streptophyta genera in winter and summer respectively, whereas Olea was the most abundant genus in spring and autumn. With regard to fungi, Botrytis and Colectotrichum were the most abundant Ascomycota genera, reaching the highest abundance in spring and summer, respectively, while Cryptococcus and Ostilago were the most abundant Basiliomycota genera and reached the highest abundance in winter and spring, respectively. The genus community structure in the PM10 samples varied day by day, and mainly along with the seasons. The impact of long-range transported air masses on the same structure was also proven. Nevertheless, rather few genera were significantly correlated with meteorological parameters and PM10 mass concentrations. The principal coordinates analysis plots and non-parametric Spearman's rank order correlation coefficients showed that the strongest correlations generally occurred between parameters reaching high abundances or values in the same season or PM10 sample. Moreover, the screening of potential pathogenic fungi allowed us to detect 7 potential pathogenic genera in our PM10 samples. We also found that, with the exception of Panicum and Fiscomitrella, all of the most abundant and pervasive identified Streptophyta genera could serve as potential sources of aeroallergens in the studied area.